Hi everyone, my name is Robert, and with this presentation I hope to show you all how we are attempting to utilize a new methodology to screen archaeal viral mutants. Alright, now the first question we usually get is why study archaea? Well, archaea branch near the root of the tree of life, and this has given us a unique window to look at how life has evolved. As you can see from the picture, they stay pretty conserved when you compare it to bacteria and eukarya. As well, their extremophilic properties can be used in industry, like their stable enzymes and such. This specific microbe we will be talking about today is called Sulfolobus, and it's indicated with the red arrow. Now, the archaeal cell we studied in the Snyder lab is known as Sulfolobus sulfatericus. The cell is often known as the E. coli of archaea because its full genome has been sequenced and its growth conditions are easily replicated in lab. Its optimal growth conditions include a temperature of 70 to 90 degrees Celsius and a pH of 1 to 4. Now, the virus that infects a cell is known as Sulfolobus turdidicosahedral virus, or STIV for short. It was originally isolated from Yellowstone National Park and is a double-stranded DNA virus. One interesting factoid about this virus is that its envelope is actually located within the protein shell, which, while not unheard of, is pretty unique. Now, one of the big reasons this virus is studied is its unique lysing system. When undergoing lysis, the virus causes 7-sided pyramids to form on the surface of the cell. These unfold like a flower to release the virions. The various stages can be seen in the picture. We know pyramid formation and lysis is caused by a single protein known as C92, as overexpression of this protein by itself will induce both in Sulfolobus. However, overexpression within E. coli only results in pyramid formation. It's theorized that C92 forms these pyramids through polymerization with itself. A theoretical model of this is shown here. Now, unfortunately, we don't really know much about the virus life cycle. We don't really know how it attaches, uncoats, how its genome replicates, or the mechanism behind its release. It's theorized that the turrets on the virus are used for attachment and injection of viral DNA, but it's currently uncertain. In order to learn more about the function of the various proteins within the virus STIV, we must first use error-prone PCR to make unbiased point mutations within a target gene. This is done out of necessity as there are no functional sites on the genes themselves. We hope that this will help us discover important codons that inhibit protein function. Simply changing a single codon is exactly what we're looking for. Unfortunately, the method we currently use to screen these viral mutants is a very long and involved process. First, we insert and grow mutated C92 into Sulfolobus sulfatericus. Once we're able to get a good amount of culture growing, around 3 to 4 days, we then split the culture into 3 and induce protein expression with a Arabinose. Time points are taken every 24 hours for 3 days. After this, we conduct an SDS page and Western blot on the time points to observe if there are any C92 protein expression. This takes an additional 1 to 2 days. We then save these time points and send them off to be imaged via electron microscopy. This process can take up to a week and a half to do, and we can only test one mutant at a time. There is a need for a more time-efficient methodology. We began with testing the yeast-2 hybrid system as a more efficient methodology. This process begins with the proteins Gal4-AD and BD. When within close proximity to each other, these proteins allow the transcription of reporter genes, which will tell us if there is protein-protein interaction. As C92 interacts with itself, attaching a copy of these proteins to Gal4-AD and BD will allow for transcription of these reporter genes. This will tell us if the C92 mutant is able to function correctly. The plasmids that encode for Gal4-AD and BD are called PGAD and PGPK respectively. To create controls for our experiment, we first had to insert SV40 large T antigen into PGAD and murine P53 into PGPK. This is our positive control, as these two proteins are known to interact. We inserted the Layman protein into another PGPK plasmid. This is the negative control, as it doesn't interact with SV40 large T antigen. Once the constructs were transformed into yeast, we grew them on the specific antibiotics that the plasmids were resistant to. We observed successful growth of each individual construct. Our next step was to mate the controls, so we mated PGAD SV40 large T antigen with both PGPK murine and Layman to create yeast with both these plasmids inserted in them. Now, both our positive and negative controls should be able to grow on plates with both tryptophan and leucine antibiotics, as the genes found for these specific resistances are within the plasmids rather than depending on an interaction between two proteins. The yeast with both SV40 large T antigen plus PGAD and murine P53 plus PGBK inserted should be able to grow blue yeast colonies on aerobasin A plates with xgal, as it is our positive control. The proteins Cal4AD and BD are able to interact due to the protein-protein interaction between SV40 large T antigen and murine P53. Our negative controls consist of SV40 large T antigen plus PGAD and lamin plus PGBK both inside a yeast cell. These two plasmids should not be able to grow on aerobasin A plates with xgal, as SV40 large T antigen doesn't interact with lamin. Thus, Cal4AD and Cal4BD are not able to interact. And that is exactly what we saw. Our positive controls produced colonies on both tryptophan-leucine plates and tryptophan-leucine xgal aerobasin A plates. Our negative control was only able to produce colonies on the tryptophan-leucine plates. It was unable to produce any colonies on the tryptophan-leucine xgal and aerobasin A plates. Because our controls worked, we then moved on to C92 insertion. This essentially followed the same procedure as before, except we used C92 instead of SV40 large T antigen, murine P53, and lamin. Once everything was inserted and transformed, we grew PGAD plus C92 and PGBK plus C92 on their respective antibiotic plates. We then mated both yeast constructs. Because C92 interacts with itself to form these pyramids, it should mirror the positive control previously shown. Unfortunately, that is not what we saw. We were able to see growth on tryptophan-leucine plates, confirming that both plasmids were present in the mated yeast colonies, but every attempt to grow yeast on tryptophan-leucine xgal aerobasin A plates yielded negative results. Multiple tests yielded the same thing. This, along with successful positive and negative controls and the growth of the tryptophan and leucine plates, told us that these proteins were not interacting with each other. Further research, however, showed us that the yeast-2 hybrid system is not the best at identifying protein-protein interactions between membrane-associated proteins, as all the work is primarily done within the nucleus of the cell. This creates issues with the membrane-associating proteins, as they now must be located to the nucleus, where they may not function correctly. Thus, we are currently in the process of testing out a new system, known as the split ubiquitin system, that keeps all the interactions located within the membrane of the yeast cell. We believe this will yield us a better alternative to the yeast-2 hybrid system, as it is specifically designed to work with membrane-associating proteins. Now, with that being said, I'd like to thank Dr. Jamie Snyder, the RISE program, Cal Poly Pomona, and everyone else in my lab. I really could not have done this without them. Here are my citations. And finally, I'd like to ask if there are any questions.